So today we're going to be looking at conception and gestation up until the creation of the placenta. So let's first start with the sperm. So let's just look at the morphology of the sperm. You've got the head, which has the genetic information, and the acrosome, which will be used for the acrosomal reaction. Midpiece contains mitochondria, produce ATP for energy, and that energy is used to power the tail. The tail is used for locomotion, movement, and the sperm become very rapid, and we'll talk about that later. But basically, sperm that leave the testes are immature, so they're only mature in the epididymis. And then half the sperm in the female reproductive tract go into the wrong fallopian tube. So half the sperm are lost by going to the wrong fallopian tube. So the first sperm is able to reach the fallopian tube within about 15 minutes after ejaculation. And sperm are able to stay and survive in the cervix for 24 to 48 hours after conception. And this is a mechanism to ensure that ovulation has taken place, and the cervix provides favorable conditions where the sperm can thrive and then eventually fertilize. If there's no ovum present, then ovulation can take place and then it's ensured to fertilize. So here's a little size comparison. There's a sperm, here's an egg. You've got two layers on the outside. You've got the corona radiata and the zona pellucida, which is just basically like a layer of glycoproteins. There's lots of mitochondria and there's genetic material in the egg as well. So let's talk about capacitation. So capacitation is the maturation of sperm. So there's two types. The first one happens in the male reproductive tract. So here, first in male. And here in the male reproductive tract, there's an increase in the motility of sperm, increase in the fertilizing ability of sperm, and losing the cytoplasmic droplets. Okay, so we've talked about this before. But then in the female reproductive tract, capacitation occurs, which is basically the hyperactivity due to influx of calcium, Ca2+. So basically what happens is that capacitation occurs due to the binding of GAC, which is glycosaminoglycans, which is highly concentrated in the cervix. And it's the role of seminal plasma, which is all of the components of semen other than sperm, to bind GAC. So other cellular changes needed for the acrosomal reaction and subsequent fertilization also occur. So the sperm membrane here, around the sperm head, becomes very fluid. So then the sperm moves and penetrates the corona radiata. So it moves through here, penetrates the corona radiata over here, while the hyperactivity occurs. So this becomes hyperactive, moves very fast, and this hyperactivity capacitation due to the influx of calcium Ca2+ allows the sperm to penetrate the corona radiata. The binding of the head of the sperm to the glycoproteins, the zona pellucida, specifically, there's a protein in the zona pellucida called the ZP3 protein. And when the sperm binds to the ZP3 protein, it triggers the acrosomal reaction. So the acrosome reaction occurs within the head of the sperm. Calcium Ca2+ is required. And basically, it's the fusion of the outer sperm membrane with the sperm plasma membrane. The formation of the vesicle, so this the hydrolytic enzymes, the digestive enzymes, which are located in the acrosome, are then released, and it's able to digest through this primary oocyte and therefore attached into the secondary oocyte. It infiltrates into this perivitelline space and reaches the plasma membrane of the secondary oocyte. So this is called the perivitelline space. You can see it here. And so the acrosomal reaction, basically, these digestive enzymes allow the sperm to digest through here, through this primary oocyte into the perivitelline space, which is this space here. And then it adheres here to the secondary oocyte, which you can see over here, the secondary oocyte. So after the acrosomal reaction, so there's adhesion of the sperm head to the secondary oocyte. There's two types of blocks to polyspermy. So what is polyspermy? Polyspermy is when more than one sperm fertilizes the egg. So we can't have that happen. So there's two blocks to polyspermy. The first one is the fast block. The fast block is basically after fusion over here, the oocyte membrane here depolarizes and it allows no other sperm to penetrate the primary oocyte. The second block to polyspermia is the slow block, and this is due to the cortical reaction and the vitelline block. So it's also stimulated by the depolarization of the oocyte membrane, but then a wave of intracellular calcium is released from the sperm head into the egg. And basically the calcium causes these cortical granules, which are located just under the secondary oocyte membrane, lets these cortical granules beneath the membrane releasing their contents. And when they release their contents, it renders ZP3 on the zona pellucida inactive. And when these parts of the zona pellucida, these ZP3 proteins are inactive, then obviously sperm cannot bind anymore. And after this, the sperm head forms a pronuclei, as does the genetic material in the egg, and these two pronuclei undergo syngamy. So syngamy is basically when the pronuclei of the male and female fuse to form the zygote. Next, we've got gestation. And in order to understand gestation, we must first understand the cleavage. So here we've got a zygote, so sperm, egg, pronuclei, syngamy form, and then it becomes a zygote when the two pronuclei fuse. Then you've got a 2-cell stadium, 8-cell stadium, you get down to here, 32 cells, which is called a morula, and then a morula differentiates into a blastocyte. The blastocyte is what implants into the uterus. And first, we have ovulation. Day 1 is fertilization. Days 2 to 4, cell division takes place in the fallopian tube. Days 4 to 5, the blastocyte reaches the uterus, and in days 5 to 9 is when the blastocyte will implant into the uterus. This differs between species. You can have cattle's take about 24 hours for one cell to divide, and then it enters the uterus in about 3.5 days. Horses enters the uterus in about 5 days, sheep 3 days, and swine 2 days. So let's talk about blastulation. How do you create this blastocyte? So basically, blastulation occurs in the morula stage, so when it's this 32 cells here. So what happens is that these cells come closer and closer together in the middle. Can you see they come closer together here? And this is called compaction. So the cells get closer together in the center of the embryo, and here on the outside you can see they're different colors. They start to differentiate. And when the cells on the outside differentiate, we call them trophoblasts. And the inside cells, they're called embryoblasts. But they don't stay like this for long. Compaction occurs to a higher degree, and then these embryoblasts all compact to one side, and that's called inner cell mass. And they leave a bit of an opening here that we call a blastocele. So the presence of the blastocele and the inner cell mass indicates that we now have what we call a blastocyte. And then at the same time, the zona pellucida, these little glycoproteins around the outside, start to disintegrate. So you can just imagine them kind of disintegrating, and that's why between here and here we lose our zona pellucida completely. A blastocyte is an embryo of 32 to 64 cells. There's an inner cell mass of embryoblasts and an outer ring of trophoblasts, and a blastocele present. So when we continue to differentiate, we've got another cavity formed here. This is called the amniotic cavity, which forms within the inner cell mass. And the bottom embryophytes here differentiate into hyphoblasts, and these ones above are then called epiblasts. Okay, so these hyphoblasts and epiblasts, let's look at them in detail. So here we've got our two germ layers, and we call these two germ layers a bilaminar disc. So here's the view of them from the side, and here's the view of them. Say we take a cross section of this is the cross section. Okay, cool. So what happens is that a layer of hyphoblasts and epiblasts is known as a bilaminar disc, and this little pink line is called the primitive streak. So the primitive streak forms in the middle of the bilaminar germ layer, and this in the middle of the bilaminar germ layer, and this indicates the start of the next phase, which is known as gastrulation. So what is the primitive streak? So the primitive streak is the site of cell migration, which we'll talk about now. Gastrulation. This is a very short process. Basically, the cells from this primitive streak start to migrate out into the bilaminar germ layer, and they start to differentiate. And these differentiations result in the creation of a trilaminar disc, 3 germ layers. The top one called the ectoderm, middle one called the mesoderm, bottom one called the endoderm. The ectoderm turns into the central nervous system, sensory organs, mammary glands, sweat glands, skin, hair, hooves, etc. Mesoderm, over here, differentiates into the circulatory system, skeletal system, muscles, reproductive system, kidneys, urinary ducts. And then the endoderm over here will differentiate into the digestive system, liver, lungs, pancreas, thyroid glands, other glands, etc. So that's basically gastrulation, just the migration of these cells from the primitive streak to create this trilaminar disc. Now we've got neurulation. So the word neurulation, you can guess something to do with the brain, neurons, notochord, etc. So neurulation is basically when in the middle of this mesoderm layer, right under where the primitive streak was, a core starts to form. These mesoderm cells in the middle start to differentiate into what is called a notochord. Notochord. Basically, the differentiation of these mesoderm cells induces a change in the above layer, in the ectoderm layer. So you can see this thickening. Let me just draw it a bit thicker. This thickening of the ectoderm layer, that is called a neural plate. So the neural plate cleaves. It cleaves into the ectoderm layer towards the notochord and creates an invagination called a neural tube. At the same time, the individual cells from the ectoderm layer break off into the mesoderm. You can see those little black spots in there. And those are known as neural crests. Now we can talk about implantation. So about a week after fertilization, you've got your blastocyte here. And so we've started losing our zona pellucida already. And this implantation takes place in the endometrial cavity. So we're in the endometrial cavity here. This is the endometrium lining right here. So the endometrial lining starts to proliferate. So what this means is like it starts forming a valley sort of thing. So it forms a valley called a crypt, which around the blastocyte, and then apposition occurs. And what apposition is the contact between the blastocyte and the endometrial lining. So this is apposition, this contact here. But eventually, the endometrial lining that's very active starts to divide and replicate, and then as well as the blastocyte starts to divide and replicate. And as you can see, these cells start to divide into the endometrial lining, while the endometrial lining starts to divide and cover the blastocyte. And this is called adhesion. So the trophoblasts start to divide and invade the endometrial tissue, and this makes sure that the blastocyte is implanted firmly into the uterus. Endometrial cells also divide and eventually engulf the full blastocyte into the endometrium. So at the same time, this collection of blood fed by blood vessels in the uterine artery are located in the endometrium as well. So they get bigger and bigger as they start to fill with blood and start to become irregular and coalesce. These trophoblasts that are dividing into the endometrium, they continue to divide, but eventually they become big and fuse. And I've drawn those big fused cells in yellow. And they become these large cell conglomerations, which are multinucleated cells, and these are called syncytiotrophoblasts. So these are called syncytiotrophoblasts, these big yellow conglomerations of trophoblasts. And the trophoblasts that do not fuse, these are known as cytotrophoblasts. So these ones that do not fuse. So syncytiotrophoblasts continue to grow and form these finger-like projections, which are called villi. At the same time, these uterine blood vessels over here, they also continue to grow and fuse. More cytotrophoblasts grow out to line the villi. As you can see, they're lining this villi that they've just created. And within this villi, you've got fetal blood vessels that start to form these little spots here. So these fetal blood vessels come into close contact with the uterine blood vessels, and they're only separated by a single layer of cytotrophoblasts. See, so there's only one cell layer between the fetal and the uterine blood vessels. So this close proximity is that so the nutrients from the uterine blood can diffuse easily into the fetal blood, And also, waste products from the fetal blood can diffuse out into the uterine blood. So this structure continues to grow until it almost covers the whole inside of the uterine cavity. So imagine the whole uterine cavity is covered with this structure, and this structure is otherwise known as the placenta. Next time we'll talk about gestation, the hormones that are required for gestation, and also parturition. Thank you.